Hello everyone, and welcome to the first recorded lecture of Anatomy and Physiology 1 online. Today we'll talk about the first chapter of the textbook, which will be an introduction to anatomy and physiology, as well as talking about Atlas A, and a little bit more about the scientific method. So what is anatomy and physiology? Right? Anatomy is the study of the structure and organization of the body. And anatomy can be divided into gross anatomy, looking at larger organs. You might do that by dissection. And also microanatomy, histology. That's the study of tissues. And for that, you would have to use a microscope, hence the microanatomy. Physiology deals with the function of the body and all of its parts. So anatomy is structure, physiology is function. And structure and function are always united in science. So anatomy and physiology complement each other and in fact cannot be divorced from one another. So structure determines function. If you disrupt the structure, you will disrupt the function. And understanding anatomy and physiology helps us stay healthy and can also help us cure diseases. So what is a disease? Right, we could say that a disease is a condition in which the body fails to function normally. And pathology is the study of disease. Every disease has a cause, and the etiology is the study of what causes a specific disease. And as we know, diseases spread, and epidemiology is the study of how diseases spread among populations. So health we could define as the condition of the normal functioning of the body. Right, free from illness or injury. So if you have proper structure, proper anatomy, you should have the right physiology, the right function. And in order for this function to be, always in balance and always regulated, there has to be the maintenance of internal conditions. So for an organism to be healthy, we need the internal conditions to be within a very narrow range. We don't want disruption of this balance, or else that can cause disease. So you might have heard this quote when you were very young. Everything exists in a delicate balance. Right? So for health, we know we need this biological balance, but in fact, everything on planet Earth exists in a delicate balance. And this quote was said by Mufasa from The Lion King. And homeostasis is a very important word. So that should be the first word you're underlining after anatomy and physiology. Homeostasis is the biological balance essential for life. So we can define it as the maintenance of a relatively stable internal environment by self-regulating physiological processes. So this is like an automatic regulating process. So it's self-regulating, it's built in. This is what allows us to function normally in the world. It is a dynamic equilibrium, meaning it's always shifting, but it's maintained through feedback mechanisms. And we'll discuss feedback mechanisms on the next slides. Disruption of homeostasis is what will lead to disorder and disease. So homeostasis is controlled by feedback mechanisms, and we have negative feedback and positive feedback. Negative feedback is the most common type of regulatory mechanism, and negative feedback keeps physiological variables within their normal range. What this means is every physiological variable has to be close to a set point. So for example, temperature, we have like a set point of approximately 98.6 degrees Fahrenheit or 37 degrees Celsius. So that's our set point. That's where our body likes to be in balance. But of course, there's natural fluctuation in temperature. So negative feedback will start when the variable is very far from the set point. So if our body temperature is very far from where it should be, that's when the negative feedback mechanism will kick in. That mechanism will then stop automatically once the set point is reached again. So again, this is like this negative feedback is self-regulating. So we'll talk about body temperature and blood pressure as examples of negative feedback. And just to show you over here, this flat line represents the set point of a certain variable. And there's always fluctuations, right? Certain things are very sensitive, so we always want it to be very, very close to the set point. Other variables might not be as sensitive, so there might be larger fluctuations over time. But before we go into actual physiological feedback mechanisms, it's very helpful to illustrate negative feedback in a thermostat, which is not alive. So we want our room to be at a nice room temperature, right? 68 degrees Fahrenheit, or let's just say let's say 20 degrees Celsius. We like our rooms to be 20 degrees Celsius. Suppose the room temperature falls to 19 degrees Celsius. Uh-oh, that's far from set point. What's going to happen is that decrease in temperature will activate the thermostat, and the thermostat will then activate the furnace. The furnace will turn on. The heat will then increase the temperature to 20 degrees. Once it reaches 20 degrees, the thermostat is programmed to shut off. Therefore, it will be back to comfortable room temperature. Once the room cools down again to 19 degrees, then this whole thing will repeat. So basically what the thermostat is doing is it's telling the furnace only to activate when necessary. And the furnace is shut off whenever it's too hot in the room. Same idea in our bodies, right? We have this set point where we want to be 37 degrees, roughly. If our body gets too hot, there's a trigger. So we have special receptors in our body that detect this increase in temperature and will cause our blood vessels to narrow. That's called vasoconstriction, right? If we get too cold, right, we start to shiver. If we get too hot, then we get vasodilation. That's when our blood vessels increase in diameter and heat can escape. So then we're going to cool off by sweating. And then we'll get back down to the set point. So again, we're always kind of fluctuating in thermoregulation. So when we're too hot, we start to sweat and our blood vessels dilate. So that will bring us down in temperature. When we get too cold, that means we are going to constrict our blood vessels. We want our blood vessels to stay closer to the internal organs, and we're going to start shivering. And that creates heat through the slight contraction of muscles. So shivering is the contraction of muscles, and that can generate heat to warm us up. One other example of negative feedback is the homeostatic compensation for postural changes in blood pressure. So when we get out of bed, we have a great shift in blood pressure, right? When we were sitting flat, when we were lying flat, blood pressure was relatively stable or horizontal. But when we stand up, the blood drains from the upper body, right? That's going to create an imbalance in homeostasis, right? All the blood, we don't want all the blood to just pool to our feet, right? That won't be good because that's what gravity wants to do. So what happens is once we stand up, special receptors called baroreceptors above the heart respond to the change in blood pressure. They detect all the blood is falling, so to speak, to the floor. And those baroreceptors of the heart will then tell the brain, and specifically the brain stem, to increase the cardiac cycle. What that means is the brain will tell the heart to beat faster. And if the heart beats faster, then the blood pressure will rise back to normal and homeostasis will be restored. So there's always a receptor, which is the structure that senses the change in the body. That could be, in our previous example, the stretch receptors above the heart, the baroreceptors that monitor blood pressure. Then there's the integrating center or the control center that makes the decision based on the sensory information. So that was the cardiac center of the brain that received the information from the heart and then made a decision to tell the effector, which happens to be the heart as well, which is the organ that carries out the final corrective action to restore homeostasis. So this was the heart increasing contractions to then increase the blood pressure. So positive feedback is not as common in biology. Positive feedback mechanisms amplify and continue the change created by the original stimulus. And this can be harmful because these cannot stop on their own. Right? Negative feedback is self-regulating. Positive feedback is amplifying. So these are used for rare events rather than daily homeostasis mechanisms. Examples of this are fever, immune response activation, blood clotting, and labor contraction. So the book goes into detail about this a little more. One example I'll talk about is in labor contractions during delivery. So when the baby is ready to be delivered, the head of the fetus pushes against the cervix. And what happens is the muscles of the cervix send a nerve impulse to the brain, specifically to the pituitary gland. And the pituitary gland figures out, right, and detects, okay, the fetus is ready to, exit. It's hitting the cervix. So let me increase the concentration of oxytocin in the bloodstream. So oxytocin is a hormone that gets released into the bloodstream, which then stimulates uterine contractions even more. And that will push the fetus toward the cervix even further. So with a little pressure against the cervix, the brain releases oxytocin, which causes the cervix to increase contractions even more. And then this happens even more and more and more until the fetus is delivered. So what are other characteristics of life? We said that homeostasis was the first one, the relatively stable conditions that all living things must maintain. But all living things have other characteristics that they share as well. One of the major ones is being made of cells. All living things are made of cells, right? Living matter is always compartmentalized into one or more cells. We'll talk a lot more about that in a future chapter. All living things are able to develop. They can differentiate and grow. All living things must be able to reproduce, meaning they produce copies of themselves, passing on their genes made of DNA to their offspring. All living things have internal chemical reactions that constitute metabolism. So these are chemical reactions, and we'll talk about this in chapter 2. All living things must be able to respond, right? They have to be able to sense and react to a stimulus. All organisms have some capability of movement, either within the organism, and all organisms can move substances throughout the body. All living things are organized, so living things exhibit a higher level of organization than non-living things. And this we'll go into detail about this in a few slides from now. And all living things evolve, right? So all living things feature changes in genes over time. And that's all that evolution is. Evolution is the change in genes over time. So the theory of evolution explains both the diversity and the unity of life. And the theory of evolution suggests how all living things descended from a common ancestor. And so we can think about this as common descent, right? We all descend from a common ancestor, but with modification. So there are changes at every step of the way. So like every other living species, we humans have been molded by millions of years of natural selection to function in our given environments. And many aspects of human anatomy and physiology reflect our ancestors' adaptations to the different changing environments. So it's important to understand our evolutionary history so we can understand why the human form is the way it is and why we function the way we do. So we can define evolution as the change in the genes of a population of organisms over time to become more suited to the environment. Key words here: evolution happens in populations of organisms over time. So no one person can evolve in a generation. So it's over many, many generations organisms evolve. And this happens because genes, which encode proteins, as you learned in biology, those determine traits. And genes randomly mutate to generate biological diversity. So different organisms will just randomly have different traits. An adaptation is any modification to a trait that makes an organism better able to function in a certain environment. So the diversity of life exists because over long periods of time, organisms respond to changing environments by developing new adaptations. And again, organisms can't choose to adapt. They automatically just randomly generate new changes, and only some of them will be beneficial. So natural selection is the mechanism proposed by Charles Darwin and Alfred Wallace. And what they proposed was that some aspect of nature selects the traits that are more beneficial to be passed on to the next generation. So natural selection means that nature, the environment, is selecting which traits among the organisms are going to be beneficial so they can survive and reproduce and pass on their genes. Right? So the adaptive trait would allow for the next generation to survive and reproduce and pass on those genes. So there are many examples of this that you've learned in biology. You might have heard of survival of the fittest or death of the weakest. It's kind of how natural selection is often framed. But I also want to shed light on it. It's not about this struggle for existence all the time. Because yes, life is tough and life has challenges, but we also depend on each other. So there's also a snuggle for existence. All organisms rely on each other to cooperate. So it's not just a struggle against each other. We also have to actually work together so we can all survive and reproduce in the biosphere of planet Earth. So humans are primates. So humans have a very interesting evolutionary story, and we can look at fossils and DNA to really shed light on where we come from. So humans are in a group of placental mammals called primates. And monkeys and apes are also primates. And about 6 million years ago, we had our last shared common ancestor with other primates. So we did not come from monkeys, but about 6 million years ago, there was an ape that gave birth to, let's say, 2 daughters, and one of those would end up being our great, 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 great grandmother, so to speak. And again, this is simplifying things, and that's really out of the scope of anatomy and physiology. But what is in the scope of anatomy and physiology are the adaptations in physiology and anatomy that are shared among primates and no other mammal. So primates are able to grasp with opposable thumbs, so we can grab onto things, unlike any other mammal. Primates have binocular vision, so we can see 3D, really good depth perception, and we have overlapping field of view. We'll talk a lot more about that in chapter 16 when we talk about vision. And most importantly, it's our large brain. So primates have the largest brain compared to other mammals. Again, there are, like, whales have very large brains as well, but primates have exceptionally large brains for their size. There is unrefutable evidence that we are related to apes, like orangutans, gorillas, and chimps. We can look at bone structure, just compare the skulls of all of these primates, right, all these great apes, right, look very similar. We can also infer certain diets. So because of what you can see certain musculature and some of the crests over here, right, indicates that there must have been a lot of muscles for chewing. So we might infer that, yeah, maybe the gorilla had a diet that had a lot of chewing. And that's true, right? We know that humans didn't require as much chewing because we invented fire. Not we, but our ancestors did. So we didn't require as much chewing because we can cook food and digest it more easily. Molecular data also supports the anatomical data. So chimpanzees are almost identical in DNA to us, even though we look very different. If you look at the chromosomes 1, 2, and 3 are almost identical amongst humans, chimps, gorillas, and orangutans. So it is pretty direct evidence that we have much in common with apes. But what is a human? So a human is a member of the genus Homo. So we are Homo sapiens, right? Wise man. But we are, I should say, we were not the only humans. Currently, we're the only humans. But we had many other human cousins, like Neanderthals and Homo erectus, right? So Homo erectus means they belonged to the human group. And species of Homo, so humans created stone tools that were very detailed. They walked upright bipedal, and they had larger brains than any other primate. It's good to know that other animals use tools too, but the human tools were very intricate, and because of their hands, they had hand dexterity, they had a lot more ability to maneuver with tools. And like I mentioned, humans were the only hominids, the only great apes that used two legs that walked upright. That's called bipedalism. So walking upright on two legs. Whereas many apes moved by swinging or brachiation, right? Like on the monkey bars. And bipedalism has some advantages, right? We can hold our tools with our two hands while walking on our 2 feet. And we can also stand up higher and search for any prey to eat or any predators to run away from. So this is a totally separate tangent that I am very interested in talking about. We said Homo sapiens is wise man. So where did the wisdom come from? So our large brains evolved, right? And there must have been, again, some pressure from the environment so that a larger brain was favored. But large brains are large drains, right? They use a lot of energy. But for some reason, I guess something in the environment said that a larger brain had some advantage. So that was selected for and allowed for very complex language and culture, which are knowledge, beliefs, and behaviors that we can pass on to the next generations. So this big brain must have been very advantageous so we can pass on these traits that can mutate a lot faster than genes, right? We have a lot more flexibility in culture than we do in genes. So we can transmit information a lot more fluidly and easily and quickly than genes. Culture is not entirely human, I can argue, right? So different species can have different traditions, different beliefs, but as far as we know, humans have the most sophisticated culture. So anatomically modern humans, meaning the ones that look like you and I, arose 200,000 years ago. So you would have seen a human that looked just like you 200,000 years ago. However, it wasn't until 70,000 years ago that we had behaviorally modern humans that acted like you and I. So humans, about 70,000 years ago, developed very flexible language in which large numbers can communicate. So we are the only species of organism, of anything, that can communicate very flexibly. We have very complicated language and in very large numbers. So for example, and this is based on a TED Talk of Yuval Noah Harari, who I am not related to, unfortunately, but I communicated with him and he wrote 3 fantastic books. I highly recommend all of them. Sapiens is the first bestseller, and it talks about the history of humankind, where we come from. And it tells us a lot about where we're going and how society acts the way it does. It's very, very good. And there's a TED Talk that gives you a summary that's found down here, so I suggest you watch that. So the cognitive evolution, we said humans develop flexible language so they can cooperate, so they can communicate, and no other species can do it like this. So primates can communicate in very complex ways, but primates can't communicate in the thousands, right? They can't really cooperate in the thousands. Ants and bees can't cooperate in the thousands, actually, but they can't do so very flexibly. They have very rigid, very minimal communication things they can do. Humans, however, can really communicate very flexibly and in very large numbers. And most importantly, only Homo sapiens can communicate about fictions, things that don't really exist. So an ape that is not a human, so let's say an orangutan, can say, oh, here is a banana, or there is a lion, but we can't talk about religion or governments or economics, right? Only humans can do that. So because of our ability to talk about fictions, things that don't really exist, it allowed us to communicate better, and communication equals cooperation. And all social mammals rely on cooperation to survive. So this communication about fictions really enabled us to survive longer, right? And again, think about this. If I can talk to you and I can convince you about heaven, and if you do something, then you can go to heaven. Or if you believe in this, then if you believe in this and I believe in this, then maybe I'm more likely to cooperate with you. I'll trust you more. So that allows for more cooperation. And again, if we cooperate, we can build cities and build governments and cure diseases a lot faster than organisms that do not cooperate. So that is a very long tangent about the evolution of humans. And this is, again, how history started. So we said living things all have homeostasis. They all evolve. And we spoke about other things like reproduction. They all have to have development. But living things are also all organized more than non-living things. So now we're going to talk about how living things are organized at each level. So what are we made of? At the simplest level, we are made of chemicals. And chemicals are non-living, and they're made up of atoms and molecules. And we'll talk about that in the next chapter. Organelles are also not alive. They're mini organs that perform critical functions for the cell, and they're surrounded by special membranes. So each organelle has its own unique properties, so they can have their own functions. Finally, we have the cellular level, and cells are the basic structural and functional units of the body. And all living things are made of cells, and a cell is the lowest level that is alive. So chemicals are found in organelles, organelles are found in cells. All right, so here we have molecules, right? Atoms are the smallest atoms, make up molecules. Molecules can be macromolecules, very large. Those would be found in organelles. Organelles are found in cells. A tissue is a group of similar cells that perform similar functions. So we have several different types of tissue, and we'll talk about those in chapter 5 more specifically. On the organ level, an organ level consists of two or more tissues that work together to perform specific functions. So for example, a tissue type is, let's take connective tissue, or let's take muscle tissue. So muscle tissue is found in many organs. So let's take the heart. The heart is an organ that is composed of many different tissue types, not only muscle tissue, but other connective tissue like fat tissues. Right? So an organ is composed of more than one type of tissue. Then we have organ systems, which is a group of organs that work together to perform specific system functions. So the heart is part of the cardiovascular system, which includes blood vessels as well, not only the heart. And we have 11 organ systems. Here's just the overview. So pause here and see how many organ systems you can name. So we have the integumentary system. We'll talk about that in chapter 6, right? I leave you to read the components and the functions if you're not familiar, but you should know this information. Right, the skeletal system, muscular system, the respiratory system, cardiovascular system, the lymphoid system. We won't be talking about these in AP1, right? Or these are AP2. Urinary system and the endocrine system, like for hormones. The nervous system we'll be discussing in this class, and the digestive system we will discuss in AP2. And then the reproductive system. And the male and female reproductive systems are considered separate technically. So once you have all those organ systems, then you can have an organism. So all of these levels, though, contribute to body function. So before, when we wanted to understand the structure and the function of the body, we had to rely on dissection and surgery. But now, luckily, we have a lot of new techniques that allowed us to look inside of the body without cutting into it. The book goes into a lot more detail, but radiology is a branch of medicine that uses imaging technology to diagnose and treat disease. So we'll talk about a couple of them just to highlight. A CT scan, computed tomography, also called a CAT scan, is a very sophisticated application of X-rays. So radiology always uses X-rays in order to visualize things. You can, again, look in the book for a little more background about that. And radiography has been around since the late 1800s. But it wasn't until recently that we have all these brand new techniques that involve X-rays, but with a lot more complexity. So a CT scan is used, and it almost takes slices. The computer analyzes certain signals from a detector, and it produces a slice of the body. And you can see a pretty sharp image compared to a conventional X-ray. So very low intensity X-rays, and the computer analysis gives you slices, so you can look at cross sections. So you can see this is just a CT scan that was taken at the level of the eyes, right? A cross section. MRI you might have heard of. And MRI is better than CT for visualizing some soft tissue. And MRI stands for magnetic resonance imaging. So it's superior quality to CT scan and no X-ray exposure required. So this uses a magnetic field in order to give the computer an idea of what tissues are there. And the MRI can see through the skull and the spine to produce nice images of the brain and the spinal cord. So it's much better than CT for looking at nervous tissue. And again, we're not using X-ray exposure, so that's really good. So it's good for soft tissue, no X-ray exposure, and a special application of MRI called fMRI can show brain activity. So this helps us understand which parts of the brain are active in real time, like with conscious thought. A PET scan is another type of technology, positron emission tomography, and this helps us analyze the metabolic state of tissue and see if the tissue is healthy. So we inject the patient with radioactively labeled glucose, and the image color shows which tissues are using the most glucose in every moment. So you can know if, basically you can see the red regions over here indicate very high metabolic rate. So you want to see where there's green, you don't want to see too much metabolic rate. You don't want to see a decrease of metabolic rate. So you can help see if the right parts of the brain are working properly. So damaged tissues also appear dark. So in this patient over here, you can see this looks like the occipital lobe, the visual center of the brain. This must have been very active for this patient. Finally, sonography, you might have heard of. It is very, very widely used. And this produces sound waves. And the signals echo back from internal organs and produce an image. So high frequency sound waves echo back from the internal organs. And this is beneficial because it avoids harmful X-rays. So especially when you're trying to image embryos or a fetus, right, you want to be able to avoid any potential damage by X-ray. So X-rays actually could damage DNA. The con is that the image is not very sharp in an ultrasound. So you might see some of these questions called rapid response questions. And when you see this, you should pause your video and try to answer as quickly as possible, just to see if you're paying attention. So pause here. And this is a question that doesn't have a very clear answer. We would say that the best answer is C, right? Organisms contain organ systems which contain organs, which contain molecules. This is not the best multiple choice question. I put this here on purpose. Certain organisms, like a bacterium, doesn't have any organs or organ systems. So this is a question that would not appear on a quiz, but this is very important to read questions very carefully. Always use process of elimination, and second guess yourself. Like if you I should say don't second guess yourself. Usually go with your gut, but double check. Always double check and make sure that the answer fits the question. So this one is very straightforward though. So which of the following contains the other three? And the answer is D. Organs contain tissues, which contain cells, which contain organelles. So I want to just quickly review some biological concepts mentioned in Chapter 1. We already spoke about anatomy and physiology being the study of structure and function respectively. And structure and function are always related. Structure determines function. Another important concept is cell theory, that all structure and function results from the activity of cells, right? So all living things have structure and function because they're made of cells. All living things evolve, so the human body is a product of evolution over millions of years. And so all of our structures and functions were selected for in past environments of our ancestors, right? Nature selected which of our traits were going to be kept to be passed on to us today. We said there's organization, different levels of complexity from atoms to organism. We spoke about homeostasis, right? We said that most physiology is trying to maintain stable conditions in the body, a biological balance. And then we didn't talk about gradients and flow, but I want to talk about this. Matter and energy tend to flow down gradients. And a gradient is just a difference from one point to another. It could be a difference in chemical concentration, a difference in charge, a difference in temperature, a difference in pressure. And again, just according to the laws of physics, matter and energy tend to flow down gradients. They go from high to low spontaneously without any energy or without any energy input. In the body, blood flows from a place of higher pressure, right, from the aorta to lower pressure. It'll go down eventually from all the arteries into the capillaries, which have the lowest pressure. That happens by itself, just by default. That's matter and energy always go by default down. If you want to go in the opposite direction or up, that's also called against the gradient, that's going to require energy, right? So to pull something up, you're going to have to exert a lot more energy than riding something down. So this applies on the cellular level as well. Normally, if I have a lot of glucose on the outside and not on the inside, glucose will go down the concentration gradient, go from high concentration to low concentration, right? Just wants to make it an equal balance. Same idea with ions. They're going to want to balance out the charge, right? So it's the same charge outside of the cell and inside of the cell. Same thing with heat and temperature. Always want to try to balance. So the warm blood, right, the heat will escape from the warm blood into the air because the air is cooler. So we're always losing heat to the environment. So again, this is how matter and energy flow down gradients. And that is the end of Chapter 1. Now I'll talk about, it's actually not the end of Chapter 1 because I'm going to talk a little bit more about the scientific method after. But while we're talking about the human body, I want to talk about Atlas A next. So let's start talking about directional terms. A directional term describes the relative position of a body part. And in order to do this, we have to use the terms referring to anatomical position. So we have to assume that the human body is in anatomical position, which means the person is upright with the arms at the sides with palms facing outward, right, facing forward. And again, feet are flat on the floor. And then with anatomical position, we're going to see that there are directional terms that come in pairs with opposite meanings. But again, you need to have, this has to be in an anatomical position. Because again, you can't just say that the sternum is above the heart or something. Because it depends if the person's standing up or lying down, right? Maybe they're upside down. But we can say that the sternum is anterior to the heart, and that's a lot more descriptive. So is it above or below? So for those we use superior, above, and inferior. So there's superior and inferior. So we could say that the heart is superior to the diaphragm, or the liver is inferior to the diaphragm. Right? Try to visualize these organs. Is it front or back? So anterior means toward the ventral side, toward the belly side, or front. Posterior means toward the back, or dorsal. There's a little bit of a difference here. Most of the time, posterior is the same thing as dorsal. The back is in the posterior, is in the back. But in some cases, for example, a cat has its belly facing the front. It's facing the floor. Sorry, we're standing up. Our bellies are facing the front. But like a cat, for example, its belly is facing the floor. So ventral is not always the same as anterior in all animals. But in the human animal, anterior and ventral are the same thing, which means toward the ventral side is front, toward the front. Posterior is toward the back. So we could say the sternum is anterior to the heart. The esophagus is posterior to the trachea. Then we have proximal and distal. So how close is it to the point of attachment to the torso? So if it's very close to the attachment to the torso, it's proximal. If it's farther away from the attachment to the torso, it's distal. So you could say the elbow is proximal, right? It's closer to the point of attachment than the wrist, right? So the elbow is proximal to the wrist, right? Because the elbow is closer to the point of attachment to the torso, which is right here. Right at the axilla, at the armpit, that's where this appendage is attaching to. So you should note that proximal and distal are usually used when talking about limbs. Right, so it's the limb's point of attachment. How far away something from the midline is either medial, which is toward the midline of the body, or lateral, away from the midline. So you could say that the heart is medial to the lungs, right? The heart is centered. It's toward the median plane compared to the lungs. But I could say that the eyes are lateral to the nose, right? They're away from the median plane. Things could also be close or far from the body's surface. So if something is very internal, we say it's deep. If something is external, it's superficial. So we could say that the skin is superficial to the muscles because the skin is external. Or you can say that the bones are deep to the muscle. So the bones are more internal than the muscle. So this is a summary slide. You must know all these terms and make sure you can test yourself. So you could be able to fill in the blank. If I say the eyes are blank to the nose, right? You should say lateral is the best answer. Another answer could be superior. But again, when I give you these choices, I'm going to make it more clear cut. And there won't be a lot of second guessing. So I'll give you another one. The sternum is blank to the heart. I said this one twice already. So this one should be easy. right? The sternum is anterior to the heart. It's in front of. So the fingers are located blank to the wrist. You can pause here. And since the fingers are farther away from the points of attachment, the fingers are distal to the wrist. The rib cage is located blank to the heart. This is anterior. The answer is C. We can also divide the body into two portions, right? The two body regions. We have the axial portion and the appendicular portion. So again, we are going to refer to two basic regions. So the axial region or the axial portion consists of head and neck. Where the appendicular portion basically refers to all the appendages and the girdles. So it's all of the appendages and the connectors. So basically, if you drew a stick figure, this is the axial skeleton, and then the shoulders and the arms, and then the pelvis and the legs, that part is the appendicular skeleton. Within those two regions, we could subdivide the body into different regions. And I want you to be familiar with a lot of these terms. You're not going to be specifically tested, but if you're going into the medical field, you will have to know all of these. And this is a very good time to get familiar with all of these terms before you're starting your study of anatomy. So if you're serious about your studies, I would really recommend taking the time to be familiar with these different terms that refer to different body regions. So many views of the body are based on real or imaginary slices called sections or planes. So a plane is an imaginary two-dimensional flat surface that marks the direction of a cut through a structure. So this is very important in order to understand the 3D structure of a sectioned object. So a section is an actual cut or slice that shows internal anatomy. But when we say a plane, that's like an imaginary flat surface. And we'll talk about sagittal planes, frontal planes, and transverse planes. So a transverse or a horizontal plane divides the body into superior and inferior portions, or top and bottom. It's also known as like a cross section. Right? A frontal plane, also known as the coronal plane, extends vertically, and it divides the body into anterior and posterior portions, front and back. The sagittal plane passes vertically through the body and divides it into right and left. The sagittal plane also can divide the body directly in the middle. We'll call that the mid-sagittal plane or the median plane. Right? The median plane divides the body into two equal halves. If you have a sagittal plane that divides the body unequally, that is called parasagittal. So it's an unequal division between right and left side. It's called parasagittal. So here we can see the medial plane, parasagittal plane is like off center, transverse is a cross section, and coronal is like the frontal plane. So which plane is a cross section of the human body? So pause here. And the answer is B, a transverse plane. It's like a cross section. So the body wall encloses multiple body cavities, and each is lined with a membrane and contains internal organs called viscera. So the body cavity are spaces that contain organs. We have two major body cavities. We could divide it into the dorsal or the posterior cavity and the ventral or anterior cavity. So those are two major, there's other ways you could divide it, but we'll talk about those two as the major two. The dorsal or the posterior cavity consists of the cranial cavity and the vertebral canal. So this is the housing of the nervous system. Right, so the dorsal cavity is basically the cranial cavity and the vertebral canal, so where the brain and spinal cord. We then have the ventral cavity, or the anterior cavity, that consists of the thoracic cavity. So the thoracic cavity contains the lungs, right? It contains the lungs, which is found in the pleural cavity, and also the pericardial cavity, which has the heart. The abdominal pelvic cavity is also in the front, so it's still considered part of the ventral cavity. And the abdominal pelvic cavity has our digestive organs like our intestines. So together, these make up the front or the ventral cavity. We have the thoracic cavity, which have our lungs and our heart. And we have the abdominal pelvic cavity, which has our digestive organs. And membranes support and protect the internal organs in the body cavities. So we have special meninges that protect the dorsal cavity. So meninges are membranes of the brain and spinal cord that protect it. We'll talk a lot more about that. Within the ventral cavity, we have serous membranes. So serous membranes are very important because they secrete a certain fluid that allows for a little lubrication. So there's not a lot of friction when internal organs rub against each other. So the serosi or serous membranes line the cavity and surfaces of the organs, and they secrete a watery serous fluid that acts as a lubricant. And we'll talk a lot more about membranes in chapter 5. So the last portion of this talk will be on the scientific method, which is part of chapter 1 in the textbook technically, but it's the last thing that we're going to talk about today. So take a second to think about what is science and who is a scientist. And the fun question is, what do children and scientists have in common? And what do they have in contrast? So pause here and tell me what you think. So on one hand, both kids and scientists are very curious. Something that they have in contrast is discipline, right? So both scientists and children are fascinated by the world around them and want to touch everything and investigate it and ask a lot of questions. Kids are just less disciplined about it. So the scientific method and science is basically disciplined curiosity. And it's really important now more than ever, especially in health science, to understand the difference between science and pseudoscience. So the discipline that accompanies a scientist's curiosity is the rigorous and creative application of the scientific method. So the scientific method involves disciplined creativity, careful observation, logical thinking, and honest analysis of one's observations and conclusions. What makes a good scientist is the ability to make insightful guesses and then imagine clever ways to see if they're true. So you, in fact, are a scientist. You do science without even knowing it, right? You make guesses about things all the time and see if you test them out. Whether it's in cooking or in grooming, we do it all the time with our pets. But we'll talk about the scientific method as a standardized series of steps used to gain new knowledge through research. This is a science's best friend, the scientific method. And it can be divided into these steps. It starts with an observation, and you ask a question. Why is this the way it is? After consulting prior knowledge, you then form a hypothesis. To form a hypothesis, you have to make predictions. Right? You make predictions because then you have to design an experiment. And with an experiment, you can collect data. And then once you collect your data, you can use statistical analysis to draw conclusions from that data. Of course, you might have to consult prior knowledge as well. And once you draw those conclusions, you're going to ask your peers in the field to confirm if this looks good. So if you're publishing something in marine biology, you're going to go to the best marine biologists, show them your data, and make sure it looks good. And if that looks good, you can publish your results for everyone to see. So we'll talk about these steps in a little more detail now. So the inductive method was first described by Francis Bacon, and that's the process of making numerous observations until one becomes confident in drawing generalizations and predictions. So we know what we know of anatomy by the inductive method, right? We can dissect bodies over and over again, and we can make conclusions based on what we see numerous times. So this is how we know what we know about anatomy. So proof in science is based on reliable observations that can be repeatedly confirmed. And it cannot be falsified by any credible observation. So meaning it's not proof if it could be falsified. If one person observes a difference, then it is no longer considered proof. It's no longer true in science. Right? So all truth is tentative, ever changing. Right? We need proof beyond a reasonable doubt in science. The other method is called the hypothetico-deductive method, and that's how most physiological knowledge was obtained. So this starts with a question. The investigator formulates a question and then comes up with a hypothesis, which is an educated speculation or a possible answer to the question. So a good hypothesis is consistent with what is already known, and a good hypothesis must be testable. A good hypothesis must also be falsifiable, meaning it must be able to be disproven. So you must be able to know and specify what evidence it would take to prove your hypothesis to be rejected. So investigators come up with a hypothesis to explain what they observe, and then they have to do an experiment to deduce whether the hypothesis is supported. So an experiment is a series of procedures to test the hypothesis using deductive reasoning, and the manner in which a scientist conducts an experiment is called experimental design. So a good experimental design really makes sure that the scientist is only looking at one factor at a time. This one factor is called the independent variable, or the experimental variable. You only want to modify one thing at a time. Right, so the independent variable is the factor that's being manipulated and tested. So a good or valid experiment should only really have one independent variable. So suppose we're doing an experiment of what dose of vaccine should be administered to babies in order to prevent a rotavirus illness. So pause here and tell me, what would the independent variable be? Right? What are we changing? What are we testing for? What we're testing is the dose of the vaccine. We're not testing the type of vaccine. We're not testing the age of the babies at which to vaccinate. We only want to test one thing at a time. So we're only manipulating the independent variable, which is the dose of the vaccine. And we want to see, right? So the dose of the vaccine is that what we're manipulating. The dependent variable is what we're measuring. So in this example, this experiment, what is being measured? It would be the number of children with the illness caused by rotavirus, right? The infection rate. So what we want to do is we manipulate the independent variable, we change the independent variable, like the dose, and then we measure the dependent variable to see what the effects are, right? So depending on the different dose of vaccine, we're going to measure the infection rate in children. And then we're going to see, does the dependent variable, does the infection rate depend on the dose? Finally, every experiment must have a lot of standardized variables, things that are maintained. So standardized variables must be held constant across all all subjects in an experiment. So that means like you want to have the age of the babies, the health of the babies all be the same to start with, right? You don't want to have 5-year-olds and 1-year-olds being tested with different doses because you might get different results, right? Similarly, you might want to have the same baseline health status of all the children you're testing. So there are a lot of considerations to take when you're designing an experiment. So sample size is one. So the number of subjects, whether they're animal or people, used in a study is called the sample size. And you want to have as large a sample size as possible because you want to control for any random chance events or any individual variations. So it's a lot better to test on like thousands of people than like 50. We also want to consider the controls. The control group consists of subjects that are just like the treatment group, but does not receive the treatment. And we need to use a control group as a comparison, right? So, for example, if you want to test the effect of garlic lowering blood cholesterol, there was a study where volunteers with high cholesterol were each given 800 milligrams of garlic powder for 4 months every single day and exhibited an average 12% reduction in cholesterol. So how do you know, though, if it was due to the garlic or if they would have just reduced their cholesterol like that anyway? So we needed to have another group of people who received no treatment. So there actually was a 3% reduction in cholesterol in the other group just by chance overall. So in the end, it actually did show that garlic was effective. There was like a 9% difference. So it seems to have made a difference, for example, but you need to have a comparison. So you need to have a control group to compare it to the treatment group. Psychosomatic effects are very real. So the mindset of a patient can actually affect the physiology very much. And you can test this by giving a placebo to a control group. So, for example, sometimes just giving a patient a pill, even if it's a sugar pill, the patient will automatically feel better just because the patient thinks they're going to get better. If the doctor says this will make you better, and the patient takes the pill, the brain is actually sometimes deceived into actually feeling better. And that's called the placebo effect. So we want to control for that in drug research, right? We want to make sure that a drug actually works because it's working physiologically and not just tricking the patient into thinking they're better. So we want to often give a control group the placebo, like a sugar pill or a saline solution, so they think they're getting it. So you can control for that and you can compare it to the treatment group, which is actually getting the effective drug. You also want to consider experimental bias. So you can avoid this. So sometimes like you don't want to know if a drug, like let's give you an example. If a drug company is making a drug, they might be biased into thinking that the treatment group is going to get better faster. Like they might bias their results to making it look like their drug works better than it really does. For that reason, we want to use a double-blind study where neither the doctor nor the patient knows who is getting the placebo and who's getting the actual treatment. So a double-blind study is the best. Also, a randomized study is great. So a double-blind randomized study is where a patient population is randomized. So it's random of who gets the treatment and who gets the placebo. And neither the doctor nor the patient knows if they're getting the treatment or the placebo. It's just like bar-coded. Finally, we need statistics. So we need to provide a statement of probability that the treatment was actually effective and not just due to random chance. So statistical tests can be applied to data to make sure that what we're seeing is due to the experimental treatment and not due to random variation. Once we complete an experiment, we collect our data, we do our statistical analysis. We can have our results be quantitative in numbers or qualitative in words. So take a second to pause here and interpret this graph. What we're looking at here is whether a low dose, medium dose, or a high dose was able to reduce the incidence of illness of rotavirus. And this graph shows either in green is any rotavirus illness, in blue is specifically for severe rotavirus illness. So I want you to pause here, take a second to really look at this graph. Does the vaccine work? And what vaccine dose would you administer to a baby that came into your clinic if you were a pediatrician? So let's interpret this graph by looking at the green first for any rotavirus illness. So what we should do is compare the low, medium, and high dose to the placebo. And what you're looking at on the y-axis, the dependent variable, is the incidence of illness, which is the cases per 100 child years. I know that's a little weird, but for example, if they looked at cases per 100 one-year-olds, they saw 25 of them with the placebo had rotavirus, but when they gave the vaccine, that got greatly reduced. So clearly the vaccine works. What vaccine dose would you give? I would give a low dose, probably, right? For severe cases, it looks like a medium or a high dose is actually better. So I might give a medium dose for severe cases. But if you don't know the severity of the case, just give a low dose. It does a very good job. In the vaccine experiment, one group of babies was injected with a placebo solution. What was the purpose of this? So pause here. And the answer is C, to compare with the babies that are vaccinated. So interpret this graph. This is a different graph. And what you're looking at is autism increasing over the years, autism diagnoses. And then what you're looking at is the sales of organic foods over the years. So what do you think? Does organic food cause autism? You should be saying no. That does not sound right. So things like this are very misleading. So you might have heard about the myth that vaccines cause autism. Again, that is a correlation, not a causation. It just happens to be that around the age that we vaccinate our children, we diagnose autism. So some parents must have correlated getting their child vaccinated with an autism diagnosis in the pediatrician office. Again, nothing to do with each other. Just because organic food has become more popular and just because we've recognized autism better and diagnosed more cases, doesn't mean the two are related. So we have to be very aware, very alert of data now more than ever. So always be cynical. So cynic stands for correlation is not a cause. And in class, if we were to discuss this, we'd have a whole conversation. But for now, I want you to understand that this means that just because two things are correlated, and even if there is a true statement, like there is an increase in autism diagnosis and there's an increase in organic food sales, doesn't mean the two are causative. Doesn't mean that organic food causes autism. For example, teens who consume more ketchup perform worse on their SATs. That's actually true. That was done by a study. How can that be true? How can that actually make sense? So again, we can have an interesting conversation in class, but pause here and try to think how this can actually be possible. So students of certain socioeconomic backgrounds oftentimes have to rely on a lot of fast food just to survive. And fast food has a lot of ketchup involved, right? And these students may not have access to the best SAT tutors and the best school districts, perhaps. So there might be a correlation between increased ketchup consumption and SAT performance, but the two have nothing to do with each other. So it is absolutely fine to have ketchup. You're not going to be damaging your brain cells or anything. But again, somebody can be exposed to this statistic and be very misled. So always be a scientist. Finally, we want to conclude with whether our hypotheses are supported or not. If the prediction happens just like we thought it would, we can say the hypothesis is supported. If not, if what we saw, if our results were not as we predicted, we have to reject the hypothesis and start from scratch. And when we have enough data to convincingly support a hypothesis, we will then submit our data to a scientific journal. And then again, like I mentioned, each submission is first reviewed by experts in the field. That's called peer review, because the scientists could then try to replicate the experiment to make sure it's verifiable and reproducible. Only if the conclusions are verified many times will the data be published for the whole world to see. And this is how we learn about the world around us, is scientists have to do their own experiments in their own laboratories, and they have to publish their data from their experiments. It's only after many, many years that textbook publishing companies compile this data into textbooks. So what we're reading right now in our textbooks was accumulated from thousands of scientific experiments done through the ages. And you have to, what we're reading now is basically 20 years old, really, when you think about it. Even though our textbook is published, has a publication date of 2021, based on these figures of when this recording, these slides and these figures are based on experiments that must have been conducted at least 20 years ago by physiologists and anatomists. Finally, I want to conclude with a couple of differentiations between what a fact is, a law, and a theory. So a scientific fact is pretty simple. A scientific fact is information that can be independently verified by any trained person. So for example, the fact that an iron deficiency leads to anemia. That's a fact. Right? So cause and effect. A law of nature, is a generalization about the way that matter and energy behave. So a law is a very concise, matter-of-fact generalization about how things work. So we have the law of gravity, right? Talks about how gravitational pull. The law of thermodynamics talks about how energy cannot be lost from the universe. It can only be converted from one form to another. Boyle's law talks about volume and pressure. So it just basically says it like it is. And these laws are results from inductive reasoning and repeated observations. So it could be written as a verbal statement, like the law of thermodynamics, right? Energy and matter cannot be created or destroyed, it can only be converted. Or it could be like a mathematical formula. Like Boyle's law is V is inversely proportional to P. A theory is different than what you'd think it is in English. So you can't say I have a theory that my car won't start because there's no gas in it. That's not what a theory is in science. A theory in science is an explanatory statement or a set of statements that is derived from many facts, many laws, and many confirmed hypotheses. So it's like a blanket overall explanation. So we have the theory of evolution, for example, summarizes everything we know of all the experiments that were done on all different types of organisms. So a theory is the purpose of a theory is to suggest where to go, where to study further with it, and to help predict what the findings should be if the theory is correct. So it suggests what we know, it summarizes what we know, and it suggests directions for future study. So the theory of, cell theory, for example, summarizes what we know about cells, but it also suggests directions for further study. It also sets the parameters for what we can discover in the future about cells. So we saw that one observation leads to a question. We might have many hypotheses within experiments can then help narrow down the hypotheses. So we might have to do many, many, many experiments to finally come up with a good hypothesis, which then comes up will make a prediction, to design many new experiments to then confirm our predictions. And we know only if the predictions are confirmed can we really say that something is true. And if somebody repeats the experiment and comes up with a different conclusion, we then say it is false, right? So science is very flexible, right? It's ever changing. And that is the foundation of anatomy and physiology. So this is the start of the semester. And next time we will discuss chapter 2, which talks about chemistry and the chemical basis of all living things.